Well, thanks, Jeremy, and I appreciate the opportunity to present this work. So just jumping right into it, why are we interested in sex? Well, for one thing, it's a key adaptive trait, centrally important from an evolutionary standpoint. Secondly, sex dimorphism is really common across a lot of different taxa, so just a couple of examples shown there with primates and songbirds. Sex determination is typically determined by sex chromosomes, fairly obviously, when there are separate sexes. Sex chromosomes are heterogametic, but they can be heterogametic in different ways, meaning that if the males have the sex-specific chromosome, it's an XY system, such as in mammals. If the females have the heterogametic or sex-specific chromosome, that's called the ZW system, and that happens to be the system in songbirds. Some of the other characteristics of sex chromosomes, I think fairly obviously, they contain sex determination loci. Perhaps less obviously, they also contain sexually antagonistic loci, meaning loci that confer different aspects of fitness or different responses to selection in the different sexes. So that's what allows the sexes to essentially specialize and also accounts for sex dimorphism. Those genes are typically in linkage disequilibrium, so the genes that cause female or male sterility are on different chromosomes, and they don't recombine, so you don't get sterile offspring. But you're also keeping those sexually antagonistic genes in the correct sex lineages. Partly as a result of the suppressed recombination, sex chromosomes tend to be really hard to assemble because they're comprised of completely divergent haplotypes or quite divergent haplotypes, and they often accumulate high repeat content. So there's a tremendous diversity of sex expression across the tree of life, and plants are unusual even in the broad sweep of biological diversity. So for one thing, if you look at the preponderance of the orange and in those circles that correspond to the gymnosperms and angiosperms, the orange and the purple, there are many more hermaphrodites in plants than in most other lineages. And at the same time, maybe 6 or 7%, depending on how you count, of angiosperm lineages are dioecious. And in fact, Dioecy, or separate sexes, has evolved hundreds, if not thousands of times in angiosperms. The second thing is most plant chromosomes are homomorphic, meaning that you can't tell the difference between the Z and the W or the X and the Y in a gross chromosome squash. And that's probably a function of the inherent dynamism of sex determination or sex chromosomes in plants. If you have rapid turnover, there just isn't really enough time for those differences to accumulate, those gross differences to accumulate between the sex chromosomes. So the system I'm going to tell you about today is the Salicaceae, with particular focus on the genera Populus and Salix. So Populus being one of the flagship organisms of the JGI, and Salix being a closely related shrub species, much more speciose genus, insect pollinated. Those two things are probably not unrelated. But both genera are dioecious, and in fact, Dioecy is a derived trait in the wider Salicaceae family, which suggests that perhaps there is a single evolution of separate sexes. So, questions we're addressing today are, first of all, how diverse are the sex determination systems in the Salicaceae? So that's both from the standpoint of whether they're XY or ZW, and also based on the location of the sex determination system or locus. And secondly, is there an underlying shared mechanism of sex determination, as you might expect if there's a single origin of Dioecy? So most of the work I'm going to tell you about is from a big collaboration funded by the NSF Dimensions of Biodiversity program that really builds off of resources that we developed as part of the Bioenergy Science Center and more recently the Center for Bioenergy Innovation, and also through community sequencing projects. So just really quickly, the methods we use. First, we had to figure out a better phylogeny for especially Salix, because Salix, again, with its hundreds of species and extensive hybridization, is kind of notoriously difficult from a systematic standpoint. So we used a sequence capture approach where we designed probes that were conserved across the family and captured sequences, basically 600 base exons across the genome, and constructed separate trees, and they came up with consensus trees. series. And then secondly, we devised a separate sex determination locus sequence capture system where we're targeting 60,000 loci that are across the entire genome. And the idea with this is to generate markers for a small number of males and females, so 24 males, 24 females, and then use association analysis essentially to map the location of the sex determination locus. And that also generates the data that we need to determine whether or not the species are XY or ZW. Okay, so just quickly, how do we determine XY or ZW? So what we do is once we've identified the loci that are associated with sex, we simply look at which sex tends to be heterozygous. And in the case of Salix purpurea, for example, you can see that the females are typically heterozygous for the loci that are associated with sex, and the males are homozygous. So that's clearly a ZW system. And for Populus trichocarpa, it's the exact opposite. The males are heterozygous for the associated loci, so it's an XY system. So here's the phylogeny. The Populus phylogeny is quite well resolved, and in fact, there's a paper in press or in review at New Phytologist right now based on whole genome resequencing for Populus, and that phylogeny closely matches the one that we got with sequence capture. The Salix phylogeny needs work still. You can see that some of the nodes have quite low support, but the basic structure of that phylogeny we think is pretty solid, especially the basal position of the tree-form willows here, Salix nigra. So this is the progress so far of determining the sex determination loci. So those arrows on the right are a species for which we've determined the sex determination system, both in terms of XY versus ZW, ZW being pink, XY being blue. And then the black arrows are still in progress. And then the chromosomal locations are on the nodes there. And a couple of take-home messages here. It looks like the base of each genus has sex determination on chromosome 7. So the tree-form willows have chromosome 7, and also Populus euphratica, the desert poplar, has chromosome 7 for its sex determination system. And then the majority of Populus are chromosome 19 for sex determination, and all of the Salix we've looked at, other than that tree-form Salix, have chromosome 15. So there are at least 2 transitions there. And then also we've detected up to 4 XYZW transitions. So at the base of each genus, it's an XY system. And then there's a ZW transition in the aspens. So Populus alba is clearly ZW. And then in Salix, there was a transition pretty early in the phylogeny to a ZW. And then all of the species we looked at so far up here have been ZW. So it looks like this is a highly dynamic system, and I expect that picture is going to get more complicated as we add more species and refine some of these associations. Okay, so that brings us to the next question, which is the one we were really able to address with the genomes that we developed through the community sequencing program, or whether or not there's a shared underlying mechanism of sex determination. So the publications so far have failed to find any shared gene content between the sex determination regions in the different species that have been characterized within Salix and Populus. And even within Populus, there was not any evidence of shared gene content. And I think that's partly just a function of the difficulty of assembling these regions because they're so complex. And so all of the analyses so far have been based on very fragmented genomes. So through the community sequencing program in collaboration with Jeremy and Jerry at Hudson Alpha, we developed a couple of willow genomes, a male and a female, with very high contiguity. And then my group used genetic mapping and also analysis of the depth of resequencing data in males and females to identify the Z and W pieces of that largely phased assembly. And then map those to a genetic map, and we're able to regenerate Z and W chromosomes separately. And the W chromosome, it turns out, is a couple of megabases larger than the Z chromosome, and both of those were annotated separately by phytosome. So here's an overview of the W chromosome. So you can see there's a large segment in the middle that's in complete linkage disequilibrium that is associated with sex. So that's our sex determination region. This bar along the bottom shows the degree to which the sequence is female-specific based on mapping reads of the male and female individual back to the W chromosome. So blacker means more female specific. There's some indication of elevated repeat content in that region and also suppressed recombination. And one of the reasons it's so large, so it occupies a pretty large fraction of the chromosome, is it overlaps with the centromere. And we know that from the presence of pericentromeric repeats in that region. Okay, so here's a little bit about the gene content with the Z chromosome on the left and the W chromosome on the, the other way around, W chromosome on the left, Z chromosome on the right. And this is based on all versus all blasts within the same genome. And this is mutual best hits. So it's basically the origin of the genes that are present on the Z and the W. And the green represents genes that are present on both. And what I want to highlight here is these blue regions. You can see the W chromosome is expanded for these autosomal translocations. And when you look in a little bit more detail about the origin of those autosomal translocations, most, or the majority, or at least the largest number, come from chromosome 19. Okay, and chromosome 19 is the sex determination region in populace. And in fact, some of those genes come exactly from the region that's associated with sex in populace. So that's our first piece of evidence that we have shared underlying mechanisms of sex determination across the two genera. All right, so when we took a closer look at those pieces of the genome that appear to be translocated from chromosome 19 to chromosome 15 or vice versa, we found that they had this really curious arrangement. So this is just mapping the W chromosome against itself, a dot plot, and those off-diagonal lines indicate inverted repeats, fairly large ones. And this is very similar to if you use the same exact method, so last Z alignments with the same settings, to the mammalian Y chromosome. And in fact, those structures, those inverted repeats, are generally referred to in the vertebrate sex determination literature as palindromic repeats. And they're apparently really functionally important. So the mammalian Y palindromes consist of, they're large, and they harbor genes that are mostly testes specific in their expression. And in fact, they are exactly the genes that confer sex determination in mammals, and they're also present in other vertebrates. It appears that the sequence identity of those genes is really high, the ones that occur within the palindromes, and that's due to intrachromosomal recombination and gene conversion. So this is seen as a way, in the absence of recombination, for gene integrity for really important, evolutionarily important genes to be maintained in the face of Muller's ratchet, or the accumulation of deleterious mutations. And now we found an analogous structure on a plant chromosome that also lacks recombination. And in fact, this has been long known in plants to occur in chromosomes that lack recombination, that being the large inverted repeat of the chloroplast, which also undergoes gene conversion and intrachromosomal recombination. So this may be a general mechanism for gene integrity to be maintained in the absence of recombination. All right, so we decided to take a closer look and see if we could determine if there was gene conversion happening within those palindromes. And the way we chose to do that was by contrasting Salix purpurea gene copies that occur in the palindromes to genes that occur in the palindromes in the closely related species that was sequenced by our Chinese collaborators, where there were only 2 copies. We don't know for sure that this is a palindromic arrangement, but what we were looking for were whether we had accumulated polymorphisms that were different between the 2 species, yet no sequence or very low sequence diversity among the genomically duplicated genes. So this is the data. The bottom just shows the copy number in the 2 species. The top shows the interspecific polymorphisms that we detected for each of the genes across the palindrome. And then these are the polymorphisms within Salix purpurea among those 4 copies of the gene across the palindromes, and also in Salix sichuensis. And you can see in the shaded region, there are no sequence differences for this gene among the 4 copies in the palindrome, but there are lots of interspecific polymorphisms. And the most parsimonious explanation for that is gene conversion. And that's just a cartoon showing the same thing with the different colored dots representing SNPs. Okay, so what is that gene? It turns out that that gene happens to be very strongly associated with sex in Populus trichocarpa. It's a type A cytokinin response regulator. In addition, that gene has very strongly sex-biased expression in Populus. And it's the only gene in the Populus balsamifera genome that is differentially methylated between males and females. The only gene. And then finally, we find that when we look specifically at that paralog that's located on the W chromosome, this is based on RNA-seq data that was again generated through the community sequencing program, we find that the gene is very strongly sex-biased in its expression in Salix. So we think that's a really, really good candidate for sex determination. So in conclusion, it looks like sex chromosomes are highly labile within the family. Lots of transitions from XY to ZW and movement of the sex determination region. I didn't show the data here, but the region looks very young in Salix as well. There's evidence for shared gene content, possibly a shared underlying mechanism of sex determination. And now what the project is looking into is whether the location and the sex determination type, so XY versus ZW, correlates with the ecological characteristics of the different species. So we're working on reference genomes for some of the other key species within the family, so stay tuned. And there should be a manuscript coming out describing this work to a journal near you in the very near future. I'd like to acknowledge all my co-authors. Maybe take questions.